So now I talked about proteins. Now we go for the characterization of viral nucleic acid or host nucleic acid. If you don't have nylon membranes or nitrocellulose membranes, you wouldn't be able to characterize the RNA by northern blot or the DNA by southern blot, or you cannot also do differential display analysis for gene expression analysis. So for this, you isolate the nucleic acid on agarose, and then you transfer into nylon, and then you hybridize with either radiolabeled probe or biotinylated probe, and then you develop your reaction with a set avidin labeled peroxidase or alkaline phosphatase, and then you further develop a reaction. If you don't have a membrane to transfer your nucleic acid into, you wouldn't be able to particularly and specifically define the viral nucleic acid or the host genome or host gene differential gene expression in response to viral infection. So again, if you were to isolate viral RNA or host DNA or host RNA, you need to have a membrane-based columns, and these are a variety of membrane-based columns, ranging from the many to midi to maxi, in according to the capacity of the column, you expect the yield of the purified nucleic acid. And if you wanted to do this for several samples simultaneously, you can use high throughput system. So this is the different stages of membrane-based isolation of nucleic acids of any pathogen. In our case, those guys who are working on corona wouldn't have been able to characterize the viral genome or sequence it if they didn't have the membrane-based columns. So the Nanocore sequencing technology or the next generation whole genome sequencing technology is based on silicon oxide or silicon nitride or graphene membranes. And you apply potential difference on the whole nucleic acid strand, and you get it passing through this membrane, and you're able to sequence as many nucleotides as you wish. So without this technology, you wouldn't have been able to monitor the nucleotide changes emerging or any mutation emerging. So you hear every day about variants of the virus. So you don't do this, you don't sequence variants on single copy number sequencing, but you need to sequence the whole genome of the virus, and you need to sequence as many variants as possible to record or figure out changes in the viral genome. So I'm going to share with you now something that is done by Dr. Dina Nadim, assistant professor in my lab, in collaboration with Professor Shaban team, with Dr. Heba, Dr. Marwa, and the rest of the team. We've been discussing possibilities for collaboration in the office of His Excellency when he was the vice president of the NRC at that time. I came to his office. I remember that day, I was just coming back from Germany after a long time, and I've been addressing possibilities for collaboration. So one of the things that jumped to our mind is the quality of the water, tap water that is coming in our laboratories, and whether this is enough to be used for biotech application and what kind of treatment that we wanted to do. I showed you at the beginning of my presentation the fancy deionizing double distilled water equipments or the molecule water equipments that we buy for a lot of money. To maintain or to change the columns, you need plenty of money. So the maintenance is the big issue, actually. It's not to buy the equipment, but to keep it running. And every 6 months, you need to change the spare, the columns. Otherwise, it's pain in the neck. It's pain in the neck of the scientists and pain in the neck of the administration of the NRC to afford the money. At that time, he was in charge of this. So I was writing in a paper that we need this, and we start to bargain how much would it cost, and at the end of the day, they buy it. But buy 10 columns for 10 equipments at once. Every 6 months, you need to change this. We were thinking of an economic way to replace this. So what they did, I don't know, you can ask Professor Shaban, Dr. Heba, or Dr. Marwa. They developed cost-effective multi-stage water purification system to replace or to compare the quality of the outcome of the outlet water from this system with the quality of the water that is supplied with molecular biology grade reagents, or with or coming from the molecule water equipment, the expensive ones. And this is the result that we could, the first comparison, the fat means the water that is coming from the tap water. The kits, that means that the molecular biology grade water that is coming with kits, with molecular biology reagents. And ours means the ones that they provided to us after this multi-stage treatment of the water. And apparently, you see the fed water is giving really nice in isolation of molecular weight DNA marker on agarose gel. But actually speaking, the water that they could prepare and provide to us was really very comparable, if not better of quality in terms of the sharpness and the obviousness of the isolated bands on the agarose gel. We thought that this is by chance. Okay, let's do something else. And at that time, we have established PCR reactions to detect whatever viral nucleic acid or bacterial nucleic acid. And we used the 3 different water samples or water sources in the PCR mixture reaction, the PCR reaction mix. And as you see, the fed water is next to the marker. Here, this faint band. Our is this fat, thick, very clear band in the molecular biology grade water that comes with the kit is a very long span. And I hope that the result is very clear. Their water works very nicely. If he knew this before being in the cabinet as a vice president, he would have avoided buying a lot of equipments, which were very costly, and he would have convinced also the president of the NRC at that time to replace all these systems by the system that he is building. And this is among the patents actually that he was describing yesterday that is applied now or under evaluation. So this is one of the 9 patent applications. So was it all? No, this is not the whole thing that I want. Sorry for having this rotating. I would have rotated this before presentation. So now I wanted to show you if we prepare growth medium of bacteria. 3 minutes. That's too many for me. I'm already done. So if you use the water to prepare growth media for bacteria, now you use the filtered water that they prepared, the fed water that is coming from the tap, and the controlled water that is provided by kits. You see that the bacteria like more their water. And if in some other case for Staph aureus, well-fed water was really astonishing, but it was really logic. Some people overuse this deionized double distilled water equipments and use it in the wrong approach or in the wrong direction, because some bacteria need actually minerals to grow, and they remove the minerals. And they think that they are doing something good for a microbiologist like Professor Shaban can tell you about that. that sometimes you don't need to remove a lot of minerals to be able to grow the bacteria. So so now I'm getting to the conclusions. He gave me 3 minutes. I'm done in 1 minute. Membrane filtration has major applications in biotechnology research. Building bridges between material scientists and biotechnologists is a must to improve and add on such applications. This can lead to developing spin-off companies which are highly welcome at the political level. طبعا حضراتكم عارفين تعظيم الشعر بتاع المنتج المحلي بتاعنا، مش محتاج اتكلم هنا بالانجليزي خالص. احنا بنقول احنا بنعمل علم. اذا ما كانش العلم بتاعنا translated into a proof that can exchange حاجه بنشتريها بالشيء الفلاني، يبقى قله العلم اللي احنا بنعمله احسن. طبعا هترجم بالعربي في الحته دي يعني عشان ما حدش يفهم في طبعا. This was something very special that I wanted to share with my people in Arabic. This can lead to developing spin-off companies which are highly welcomed at the political level. Any products in this direction can be absorbed by the Egyptian biotech market, by the way. I'm not talking only about the Egyptian biotech market, but we can satisfy the whole region if we really build this collaborative networks. So at the end of my presentation, I'd like to acknowledge a couple of people from my group, Dr. Dina Nadim and Associate Researcher Rula Nadim. And I wish to also thank all the team of the flat sheet membranes for the collaboration. And I thank you for the invitation. And I thank you all for your attention. And I look forward to discussion. Thank you so much.
Thank you, Mahmoud. Discussion and this application of membrane and plus opportunity to live in an area new for membrane. Thanks for the
First, I'd like to thank you to give me the chance to introduce Dr. Sayeda's presentation. This presentation for the entitled Egypt's water challenges, current situation and future perspective. This work was performed with Dr. Sayed Abdul, Dr. Haladoma, and Dr. Rabil Ali from the Water Pollution Research Department, National Research Center. Now we will transform from the virus and this complicated subject to another subject of using membrane in sub-technologies to produce products, help in medical products. Now we will see how the algae can contribute in water treatment, in wastewater treatment, and in producing microalgae for other byproducts. Water scarcity and insecurity. The notion of water is plentiful. It covers 70% of the planet is false, as only 2.5% of all water is fresh water and 97.5% is saline water. Up to 40% of the world's population will be living in seriously water-stressed area by 2035. Desalination is a major technology that has been employed to remove salts from seawater, brackish water, and saline groundwater. It is energy intensively costly and creates significant environmental impacts. Often it is employed by regions and nations with access to plenty of energy, such as Middle East and other wealthy nations. Wastewater treatment technologies. The wastewater treatment has been considered to be an essential and a challenging part that can contribute in reuse of water resources. This slide exhibit the last statistical wastewater treatment in Egypt. We produce annually 6.5 billion meter cube per year. And unfortunately, the treated wastewater is 3.7 billion meter cube, and untreated is 2.9 billion meter cube. 20% of the treated wastewater is primary treatment, and 44% of the primary treated used to cultivate forests, and 80% is secondary, and 26% of this secondary treated effluent used for crop irrigation. And 0.7 billion meter cube per year is reused, just 0.7 billion meter cube per year. The wastewater used in Egypt is an old practice. It is constructed from 1930 as Jabal al-Asfar and Abu Rawash. The most popular treatment technology used in Egypt is about 75% of activated sludge, and 11% of the stabilization bones, and 4.5% of the treating filters. We will focus on the stabilization bones. The stabilization bones steps are the anaerobic treatment that focus on the anaerobic dealing of the microorganisms. Then followed by the facultative bone with depth about 3 meters. The anaerobic bone of depth about from 4 to 5 meters. The facultative bone deal with anaerobic and aerobic treatment of the organisms, followed by the maturation bone that of larger scale with the depth about 1.5 meters. This is the most effective treatment system in Egypt, but we use about 11% of the technologies in the waste stabilization. It has some advantages and some disadvantages. The merits of this system is the low construction cost, low energy requirements, low sludge production of useful algal biomass, the greenhouse gas reduction, since algae wastewater treatment produce carbon dioxide, but also it consumes a lot of carbon dioxide in photosynthesis. So the result, the final result is for the entire system is carbon negative. So it is effective, but it has some disadvantage. It needs a large land usage, low control options, operation dependent on climate, and some other disadvantage. So our vision is to upgrade this stabilization bond. And the team of Dr. Galina began in 2012 to upgrade this system. It constructed, as we mentioned before, for the anaerobic bond, followed by the facultative and the maturation bond. The maturation bond is converted to the high-rate algal bond with another depth. The maturation bond was 1.5 meter depth, but the high-rate algal bond is only 0.5. In this system, the team produces algae and treated wastewater. However, they upgrade in 2017 to a high-rate algal bond 6 meter cube, and they cancelled the anaerobic bond and focused on the facultative bond with larger depth. And they upgraded it in 2020 with a high-rate algal bond 25 meter cube. And it covered anaerobic bond. So let's see these systems. This is the first step was constructed in 2012. This is the maturation bond, or the upgraded maturation bond, is the high rate algal bond of 1 meter. And this is what constructed in 2017. We can see here the maturation bond is converted to a high rate algal bond, and we can see. I'd like to show you here because you fish notes. Okay. Okay. Please, it's okay. The mouse is out of sorts. Okay. Now we begin here with the facultative mode in this system, where the organic compound is decomposed, and the upper stream it is decomposed anaerobically. Then the upper stream is fed to the high rate algal bond, and after a certain retention time, the upper stream is moving continuously to the settlers or the rock filter system to filtrate the water and get the algae. And where the rock filter in upgrade with Dr. Ahmed Shaban's team has upgraded to use the membrane technology here. This block column is a membrane that filtrates the water, the upper stream of the continuous flow of the high rate algal bond to produce water for users, for coded uses, coded irrigation, not for restricted irrigation, but for unrestricted irrigation and for human utilities. In this system, we have a sun dryer, so we decrease the energy to the minimum, and we will see the potentiality of algae in next slides. Here is the third one, the third step that constructed in 2020, the high rate algal bond of 26 meter cube. Now let's discuss why we upgraded the maturation bond. We have upgraded the maturation bond to increase the quality of the treated water to be used in unrestricted irrigation and in human utilities by using different methods and different technologies like the rock filter, OMV, filtration, and membranes with Dr. Ahmed. And in the same time, we have the algal biomass. The algal biomass is potential for several industries. Firstly, the first branch is the energy source. The energy source where the biodiesel, bioethanol, and bioelectricity. And the other branch is the added value. These added value components is the high value pharmaceutical compounds and the food and supplements for human or for fish and poultry. The wastewater reuse, this slide represents how much we gain from this treatment. We got algae that is fertilized with agriculture. And you can see here the wheat spice full in this picture, full of seeds. It was wonderful to use it in agriculture as a fertilizer, and the water, the treated water was used in irrigation. The second potentiality of the algal product was the extracting oil to produce biodiesel and the bioelectricity using the biofuel cells, and the bioethanol is produced by fermentation of ethanol. And it was a good experiment because I did it. The bioethanol fermentation was produced in high rate. It was 2 grams per liter. Now is great. Tell him, doctor. Pharmaceutical evaluation of algal biomass collected from high rate algal. This used for treating mouses, algal biomass, and antibiotic agent as antibiotic agent. The results was very important. It decreased glucose by 83%, increased insulin by 2.44, increased gluten 1 by 1.1 fold, and the algal biomass has anti-inflammatory. A trial for application of microalgae biomass in broilers nutrition against challenging with the Newcastle disease virus genotype 7. Normal viability of algae treated broilers, the chicken, after challenging with Newcastle disease virus, and good appetite for algae treated broiler chicken was significant. And it was examined also, tested also for the fish. The growth performance of Nile tilapia diet supplement with different algal biomass concentrations of algae dominated with microcyst species. The diet supplementation with the algal biomass collected from high rate algal bond at 5 grams per kilogram for 30 days enhances the hematological and immunity parameters in the fish and increase their resistance against hydrophilia infection. The valuable molecules from microalgae. We can find carotenoids. The first one is beta-carotene, and the astaxanthin, the zeaxanthin, and echinol. All these products are antioxidants, and they protect for the zeaxanthins, protect eyes and improve vision. We can find also the chlorophyll A that is potential anti-mutagen and anti-carcinogen. We can find also phycocyanine and hydroxyfluoride. The last slide is about the polysaccharides. Currently being investigated for its use as a joint lubricant in intra-articular injection. And the bioplastic could be produced, and it is under investigation and under test till now, is produced from algae, because we find the polyhydroxybutyrate as a derivative of the polyhydroxyalkanate was detected in algal biomass dominated with microcyst species that is found naturally in the high rate algal bond. It is now under investigation for its utilization for the preparation of surgical sutures and scaffold. Thank you. Questions?
Thank you, Dr. Sarah, for this presentation.
Thank you. It's for Dr. Sayeda, not me.
For all of you, for you and Dr. Sayeda, Dr. Camila, the whole team, and thank you for this team give us opportunity to applicate our membrane in different fields, in other fields, as tertiary, I showed yesterday, as a tertiary treatment, a tertiary wastewater treatment, thanks to you and your group to give us this opportunity for this time. And any question? So,
I'm asking about
Insulin wastewater treatment plant.
Yes. Also, I'm asking about this report for the different food and product.
Insulin. There is area, enough area for the prevention inside the stage. Both right, feed, membrane, reduce, application to the cancer, was done in the lead. And back to the middle. Another one and nothing. The small one. You mean that the pipe? You mean that the pipe in NRC, one cubic meter. When you go to large, when I am with my our membrane in your system, in the lead. This was question of Professor.
I think the question has got different part. What is the composition of the inlet and outlet? He wanted to know the composition before and after. This is what we want.
She said, unrestricted. I get it. After membrane, unrestricted irrigation. Before the membrane is a secondary treatment certification. You know the clear قرار كمان دي صفحه صفحه 100 بتاع دي secondary treatment بالكاركترستكس بتاع secondary treatment. عشان كده اسمها التعليمات اللي كانت عليها تيرشن لان احنا محللنا الميه الثانيه اكثر من التيرشن. ها؟ كل المحاضرات. اخر سؤال اتفضل.
هل في بيتا كبير؟
ايه ده؟
بيتا كبير اسمها معموله في مصر هنا؟
في بيتا بالظبط يا
دكتور.
عايز ابقى في المنطقه وعايز الريزلت بتاعت النتائج علشان في بيتا موجوده من 2010 انجلين الراجل بتاع التركيه وقدر التعامل معاهم نلاقي الانفورميشن دي هنا ممكن نستخدم الحاجات دي فعلا تدينا بقى.
باي ذا واي الصوره
انا عايز ده وعايز ده.
كل حاجه موجوده دكتوره كريم حسين علي بكل التفاصيل واستاذ التحالف في المركز القومي للبحوث وليها شرف ان هي مساهمه معاها كبيره جدا وانا عارف ان هي كل حاجه عندها دوكيومنت.
يعني طلع عشان مش هتعامل معانا بره فيه.
هو كده الدكتور المحور الاول هو كان
لان الناس جايبين الجيترز بتاعتهم
احنا
جايبين الداتا
الجيتر والشغل واحنا جاهزين ان شاء الله
ويوصلوا عروسه بايته.
Thank you, Dr. Samar.
Thank you.
Dr. Mahmoud, we'll move to the next speaker. Dr. Shaban has got 20 minutes.
I talk about antiviral membrane production and applications in wastewater treatment. First, we all know that the world has many places that suffer from drinking water pollution, which leads to the spread of serious diseases that kill human beings.
Contaminated water can transmit diseases such as cholera, dysentery, typhoid, and virus. Cases of contaminated water may be a natural contamination like climate change due to high temperature or increasing salinity. Another kind is a chemical pollution, such as as a result of mixing potable water with leaks from sewage wastewater, from agriculture or industrial wastewater. Most of this happens in remote villages and areas remote from services. Wastewater contains 10 million viruses in addition to million bacteria. Most common and dangerous viruses in contaminated water, rotaviruses, which is has the largest diameter. It causes inflammation of the stomach and the intestines. Itchy virus, 30 nanometer in diameter, can result in stomach flu. A human rotavirus has a diameter 26 nanometer, can cause severe acute respiratory infections. Can we drink safe water? Yes, by using ultrafiltration membrane. Ultrafiltration membrane usually used for water purification or wastewater treatment, industrial or agriculture or sewage wastewater treatment. In the common use in the world, they use ultrafiltration as in door system micro or MBR membrane bioreactor or outdoor membrane bioreactor. Membrane bioreactor indoor system means that membrane submerged in the biological tank, where outdoor membrane bioreactor, the biological treatment were carried out in the biological tank. Then the treated water flow to the ultrafiltration spiral wound membranes or hollow fiber membranes. So we can see that the membrane technology is the best technology to remove viruses without using any chemicals. We know all the famous membrane type. Microfiltration membranes can remove bacteria and parasites particles. Ultrafiltration usually were used for high molecular substances and viruses and protein removal. And nanofiltration can remove mid-sized organic substances and charged ions. Particles are all usually used for to remove single charged ions. What is the antiviral membrane? They are membranes that have the ability to separate viruses either by effect of small membrane wounds, which cause in a mechanism such exclusion, or through the absorption or desorption of the protein of the virus on the surface of the membrane, or using mechanism which is called electrostatic mechanism, electrostatic repulsion by membrane, charged membrane. First, most viruses in the wastewater has a range from 20 nanometer to 200 nanometer. Based on the type of this virus, the membranes, depending on the pore size, the viruses which has the largest membrane than the pore, it can be separated by the mechanism of the size exclusion. However, the charged membranes, which has a negative charge, can separate the negative charge viruses depending on the charge of the protein. Also, hydrophobic surface of the membrane can make the prevention of the viruses to pass the membrane. Hydrophobic membranes means the surface cannot pass the water, just passes the water below. How can I produce antiviral membrane? In our cases, in our experimental work, we found that the materials called manganese acetyl acetonide. These materials is metallo-organic nanoparticles. In some researches, we found that some researchers using metallo-organic nanoparticles has a manganese in its structure. These using these materials to destroy the protein layer in influenza virus. So we using the related materials of these nanomaterials, and we added it in the polymeric solution. The method of the preparation was adding this nanoparticles in the solvent, then we adding the polymer, then mixing for 24 hours under vacuum, then casting steps on an anaerobic support, and coagulation in the water bath. These nanoparticles has a negative charge, so we know that this membrane maybe contains a negative charge on the cells. We produced 4 kinds of the membranes of ultrafiltration membranes, depending on the particle size or the nanoparticles percentage addition. UF1, which is there is no any addition of the nanoparticles, has a hydrophobic surface, which the contact angle was 95 and has a particle size of 80 nanometers. Increasing the nanoparticles percent to 1.2 leads to reduction in bore size and reduction in the contact angles. That means that the hydrophilicity increased. We applied these negative charged membranes on separation of bovine serum solutions, sodium alginate solutions, and we using real soil wastewater solutions. The results found that the separation of bovine serums reached to 95.5% using UF3, which we use nanoparticles about 1% percentage in 1%. The productivity increased when we increasing the hydrophilicity. That means that ultrafiltration number 4, which has 1.2% nanoparticles, has the productivity more than UV. So about application on the separation of solution of sodium alginate, we found that U4 provides high permeate flux according to high hydrophilicity of this membrane, while U3 provides 99% during operation time. We repeated the experiments for many cycles in removing sodium alginate to indicate this membrane or to indicate the fouling of this membrane. So we repeated these experiments for 5 cycles in 10 days, and we found that the sodium alginate rejection reached to 99% after each after every washing step. So this membrane can be we can be considered as anti-fouling membranes. Another application in the sewage wastewater application, we have a layer wastewater samples and applied these samples on the membrane separation. We found that UF3 provides the good separation for most of parameters like turbidity and COD and DOD. Also, the coliform bacteria not detection in the product. Also, the virus, rotavirus, IgG virus, and rotavirus, when it was positive detection in the field, but after the membrane, it was a negative detection. About fouling experiments, we repeat these experiments more times and for long times, for a month approximately. UF3 provides high recovery flux ratio, reached to 99%, and it has low reversible resistance and low irreversible resistance. Reversible resistance means that these membranes can carry a cake of the foulant without penetration the pores. So I can clean this membrane easily by backwash. While reversible resistance makes a problem because it can penetrate the pores. That means chemical cleaning to remove the foulant from the pores. After we select the best kind of membrane, we make this membrane as a pilot scale and produce large-scale flat sheet membrane by using our continuous casting machine in our lab. This is the casting knife, and we putting polymer solution in this casting knife and drawing the flat sheet membrane and get the rolled from flat sheet membrane. We using fabricated spiral wall membrane to produce the large filter or large ultrafiltration filters. We have a UF pilot plant in Zenin station. We're using it as a secondary, as a tertiary treatment after secondary treatment by alcohol. This is our pilot plant. We use the spiral wall module 4040. It's fabricated in our labs. And multimedia just to make a water treatment after algae. We make 3 runs. This experiment was carried out for 6 months. We found that the productivity increased and enhanced during operation to reach to 36 meter cube per day. We also found the detection of virus after UF module is negative. There is no virus after filtration membrane. The negative, we resulted from that. The negative charge of virus and the membrane surface leads to the electrostatic repulsion, leading to the prevention of the virus to contact the membrane surface and come out to intake. Also, viruses are found in groups, so it can separate it easily by site exclusion mechanism by membranes. This is the result after tertiary treatment by UF membrane. We found the durability is reduced to 1.5 from 12.5. The COD and PB reduced. Also, the algae after UF membrane, no blue algae was found. Also, total algae account separated about 97%. This is the products. The first one is green one is feed. Last one is the backwash of the membrane, and this is the produced after tertiary treatment for 4 hours. Conclusion: the antiviral membranes were prepared using metallo-organic phenol particles. The membranes have small pore size, so they have 2 ways to separate the viruses: electrostatic function and site exclusion. Prepared membranes have a negative surface charge, can separate viruses with the protein negative charge, so it leads the prevention of cake formation on the membrane surface. The spiral wall module UF prepared using the membrane type provided productivity of 36 meter cube per day and enhanced during operation. Our prepared UF module can be used for tertiary treatment of sewage wastewater treatment and can be used for purification of contaminated potable water. Thank you.
Very interesting results. We need to further discuss how to confirm whether the filter viruses are alive. Okay. In what kind of viruses do you filter on the top of the membrane? So
separation from protein. So benzene and sodium alginate. Okay. About detection of the virus, we not detect one, not detect anyone, but we using the regular sewage wastewater and make analysis. We found this sewage wastewater, these three types: rotavirus and virus and human rotavirus. We just don't know account, just if it is a positive or negative.
Thank you. Any questions? Please.
البرسنت كان في ان عندك من 1 ونص و1 1.2%. 80 دي نمبر بورصه. فور سايز اوف مليون. 80 نانو 80 نانو دي بورصه.
Any more questions?
The ratio between the proportion and the product. I don't can need to say what
The ratio between the flux.
Okay. The bioflux, we work it in a bioflux for 4 hours. Okay. Each hour we make a for 15 minutes.
سؤال هو الكميه اللي تحتيها في الباكوش قد ايه من المنتج؟ مش عارف. The ratio between the used water in backwash and in the produce.
يعني الكميه المنتجه
اللي
ولا عايز
بينتج في 4
انا
ساعات.
انتجت مثلا ايوه ما هي دلوقتي انا عندي المنتج كله بيبقى مثلا 36 مكعب في اليوم. تمام. احنا لو جينا حسبناه وانا بعمل تقريبا باك ووش مثلا من ساعه او ساعتين على حسب انا بعملها كل ما الفلكس دي. كميه الميه اللي هي بيحصلها من الباك ووش دي انا في الحقيقه اصلا ما برميهاش. يعني انا دلوقتي السيستم اللي معمول ده معمول كونكتد للار دي بول. فانا دلوقتي الالترا فلتريشن بيشيل عليه ارضي. انا ما برميش ده بالنسبه لي انا مش ويست. باخد الارضي بقى برجعه تاني للارضي دي هم بيستفيدوا بيه. تمام. فانا بالنسبه لي هنا مش ويست. انا ما عملتهاش على
ما اتحسبتش لان
ما
هي
اتحسبتش. انا بالنسبه لي ما حسبتهاش لان انا مش عاملاها كويست.
I'm sorry, I have to just stop the discussion at this stage. We need to move to the next speaker. We're happy to have engineer Ahmed, chemical engineering, in a scholarship holder from CUT China. We will talk about membrane application in hemodialysis. The floor is yours.
Yes, please.
Or whatever you want. If you want to come here.
No, I.
What?
Hello, everybody. I'm so happy and so honored to be here and to take part of this important conference. But I will just take you to a journey about the medical application and hemodialysis. But before I start, I want to thank Dr. Heba very much for the invitation and for the useful guide. So I will start my talk today about talking about the hemodialysis. As we know that hemodialysis is a process of purifying blood from impurities and wastes that can exist in blood, and it's so harmful. So when there's someone with kidney failure, he results to hemodialysis to make this process through a machine called the hemodialyzer, and this machine consists of some fitted membranes that can mimic the functions of the native broken kidney, and this membrane is my talk today, and I'm gonna talk about the developments that happened to the hemodialyzer since the first time it was used to the present and the future trends. So let's start with a good question. Why developing membrane technology used in hemodialysis is an urgent field? If you don't know, but there's 10% of the global population is suffering all from kidney failure. And since it's not affordable to most of them to resort to the kidney transplant, so from here, hemodialysis application technology has emerged and has been the common trend and the medical applications and the membrane medical applications, and that's what attracted me to this field and led me to do this review article. So first of all, in the very beginning, scientists used the cellulosic membrane in hemodialyzer, and from its name, it consists of cellulosic membranes fitted together, manufactured from a saccharide called cellulose, sorry. And it was used for a long time, but because owing to its hydrogen construction, because it enabled the low wall thickness of the hemodialyzer to be combined with high porosity, and that enabled the high transport and effective expulsion of the blood impurities. But then it was no longer used because it has low biocompatibility. And if anyone here doesn't know what biocompatibility means, it is the property of a material to be compatible with the living tissue. So it was a pivotal reason to no longer use this kind of membrane. And then scientists made some more modifications on this kind of membrane to solve these demerits, but it's still not used because it has a very, very bad disadvantage, which is it couldn't filter the medium-sized impurities that could be found in blood. Later on, scientists created the scientific membranes, and this membrane will outnumber the cellulosic membranes in many things like biocompatibility and filtration capability, but it's still not the perfect and the ideal membranes because many of the hemodialysis patients who were cured with this membrane experienced oxidative stress and low blood pressure while they were treated with this kind of membrane. After that, in the meantime, researchers are focusing on reducing the usage of fresh water in hemodialysis, and also they are focusing on getting rid of the disadvantages of the synthetic membranes. And also the very, very, very new trend is that there are a group of Iranian researchers that could experiment for the first time the usage of the electrospun nanofibers membranes in hemodialysis, and they found so promising results as they found that it's as commercial as the synthetic membranes to be used in the hemodialysis, and also it has the same disadvantages of the synthetic membranes. Finally, I think no doubt that every effort could be put in this field is worthwhile due to the big amount of the kidney failure patients and due to the importance of this field. So it was my last part of my review article. So you can see in the proceeding of the conference what I have reached. I made a review article about the developments and what happened all the years from the first time statistics resort to hemodialysis and the applications of the membranes used in this field from the first time until to date. Okay, and thank you for this great event, and thank you for listening to me. And if there are any questions, go on.
Thank you so much for the very interesting and very enthusiastic presentation. We're getting to the end of this session, so unless there are no questions. Is there any question? Any question? Further. Lamir. Right, I thought that you had a question. I thought I had a question. Yes. Further. We stopped at the level of cellulose membrane for the hemodialysis. We not go with polyamide membrane.
Anyway, I was focused in my lecture on my talk, so I didn't get that important to my. Anyway, I was focused much more on my talk in this lecture, so I can take a quick glance at my PPT and show you what I have prepared. Okay, here was the first link, Muhammad Hassan Ayan, a Kenyan engineering student, and it's a picture of the hemodialysis process. As we see here, that is the hemodialyzer. Here, that's the hemodialyzer, and it just mimics the functions of the failure broken kidney in regards of verifying the blood from impurities and wastes and so on. And then here I was talking about how this
السؤال هو هيقول الست الدكتور بيسال السلوز اسيد بطلوا يستخدموه في الدياليسيس. ايه الجديد؟
What's new is the using of the synthetic membrane owing to its high biocompatibility. Here I'm showing the advantages and disadvantages of the synthetic membranes. Is they're used nowadays, and most researchers are بالظبط.
هو بقول ما احنا عايزين نقول كده. الدكتور جمال بيسال سؤال.
So nowadays, researchers are focusing on solving the disadvantages of the synthetic membranes. And here I'm showing the disadvantages and the advantages of the synthetic membranes, and also the electrospun nanofibers, electrospun nanofibers membranes are used nowadays, and it was so recent to be experimented in Iran by these researchers I mentioned, and it got so good. Promising results, so it can be the future trend in this field. Here I'm saying about what's the future trends. So as you see here, reducing the usage of fresh water and hemodialysis, recycling hemodialyzers because According to the Guardian journal, there are about 600,000 of hazardous wastes are produced as a result of eradicating the dialyzers. So it's a future trend too, and tackling the disadvantages of synthetic membranes, as well as experimenting with the electrospun nanofibers membranes in hemodialysis. So that's all.
Okay.
Okay, thank you.
هايل وبشجعك تاني